Features of adaptive immunity. These are specificity, diversity, memory, homeostasis, and discrimination between self and non-self. What is specificity? That body can recognize different types with the antigenic peptide called epitopes, and this specificity is due to the unique receptor present on the lymphocyte and which is present before the host encounters the antigen. So it is a predetermined genetic factor, this is specificity. Now the second one is diversity, and that is the host can recognize millions of microbes. It can recognize 10 to the power 9 to the 10 to the power 11 antigenic epitopes, and this is generated by the extensive rearrangement of genes for T and B cell receptors. You see, there are different types of pre-existing immune cells, A, B, C, D, and so on. Now the body encounters the antigen G. The receptor present on the G immune cell recognize this antigen and respond, and now the proliferation of the same type of clone cell takes place, and same types of the cells are produced with the same type of antibody that is anti-G, and this is the proliferation of cloned immune cells. Now the memory, the host can remember an earlier exposure for its lifetime. When the next exposure takes place to the same antigen, then response is very heightened and of better quality and which is quickly generated. And this feature of adaptive immunity is the basis for the production of vaccine. The both B and T cells generate memory. B memory cells choose the class of antibodies, and T memory cells quickly migrate to the site of infection. You see, here is the exposure to antigen A. Antibodies to A is produced on the 7th, 6th day, and but of its magnitude is lower. But when the second exposure to antigen A takes place, the response is very high. The concentration of antibodies is very high, and it is for longer duration. And again, if there is antigen B, then antibodies to the B antigen is produced with the lower concentration. Now homeostasis, and this is the when the infection is eliminated, the immune system comes to a steady state, and this time immune system switched off, secretion of cytokines is stopped, and activated lymphocytes are face the apoptosis. Discrimination between self and non-self, own body tissue or foreign tissue. That antigen receptor genes rearrangement in lymphocytes can also generate lymphocytes bearing receptors for self antigens, and when they escapes and circulate in the body, they destroy the healthy cells bearing the self antigen, and this phenomena is known as autoimmunity. Elimination of these lymphocytes bearing the receptors for self antigen is done during maturation of the T and B lymphocytes in thymus and bone marrow, but some of them again escapes the elimination and produces autoimmune diseases. Response of adaptive immune system. You know, lymphocytes express receptors for distinct antigenic epitopes. Few lymphocytes in circulation bears the receptors for the given antigenic epitope, and binding of antigen to specific receptors triggers the proliferation of lymphocyte clone. Expansion of the population of B cells, plasma cells producing antibodies, and memory cells. And the expanded T cell population produces cytotoxic effects by the T helper cells. Some of the T and B population cells go to the GO phase of the cell cycle and remain dormant as a memory cell until the next time the antigen attacks and the same attack with the same antigen. Here antigen selection takes place, then proliferation to B cells and T cells, differentiation, the plasma cells they produce antibodies, and memory cells they remain for the future infection. Here the T cell antigen binding antigen producing cells, then MHC2 interaction, T helper cell, CD4 cell, CD4 cells activate the B cells to produce antibodies, and T helper cell CD4 cells produces macrophages and CD8, that is killer T cells, cytotoxic cells. Phases of adaptive immune system: recognition phase, they identify the antigen; activation and proliferation phase, CD4 positive cells recognize and they proliferate into plasma cells and cytotoxic cells, B cells and T cells. Effector phase, antibodies and effector cells get rid of the invading microbes and some of them turn into memory cells. The last phase, they come to the homeostasis state and activated plasma cells die and activated T cells apoptosis. Entry of microbes happens through skin, respiratory and GI system, phagocytic cells, influx of phagocytic cells at the site of infection, causes inflammation, attracts macrophages, neutrophils, natural killer cells as a first line of defense. Then trafficking of cells is by the help of chemokines carried to regional lymph nodes and spleen, and the antigen is turned into small peptides and associated with MHC2 molecules and produced to the TH cells. Here recognition cell, then clonal activation phase, clonal expansion and differentiation, then effector phase, elimination of antigen by humoral immunity and cell mediated immunity, and then decline, that is homeostasis phase, that is apoptosis takes place, and finally some cells of T population and B population turn into memory cells. And the duration you see from 0 day to the day 30, and days after the antigen exposure it happens. Thank you. Be careful, wear mask, take care of yourself and your family and friends. We will meet again. Thank you.